Hello, and thank you for taking the time to check out this presentation about Tinea corporis, or more commonly known as ringworm. This presentation is a breakdown of a review article that was written in October 2024 called A Clinical and Mycological Study of Tinea corporis, a Hospital-Based Study. The authors are Sweta S. and Lakshmi H., both assistant professors, and Dr. Dilip Kumar N.R., associate professor of the Department of Dermatology, Venereology, and Leprosy at the Sri Atal Bihari Vajpayee Medical College and Research Institute. Next, we will look at the first page of the review article I'm going to discuss. It includes the title, the author's location, abstract, materials and methods, results, conclusion, keywords, and the beginning of the introduction. The purpose of this study was undertaken to find out the clinical patterns of Tinea corporis and to find out the fungal species affecting Tinea corporis in Bengaluru. While it's not exactly stated that the researchers of the study had a hypothesis, it is pretty clear that they had a purpose of what they were studying. They wanted a better understanding of which organism was responsible and what the symptoms and patterns that the patients were presenting. Let's talk about some background information from this article to understand further. What is it? Tinea corporis, a superficial fungal infection caused by dermatophytes Trichophyton, Microsporum, and Epidermophyton. This study observed 100 patients presenting with visible skin lesions distinctive of T. corporis, ages 0 to 60 in both male and female. How did they get it? Most likely through contaminated skin-to-skin contact, skin to contaminated surface, or skin to infected pets or animals. And where is it most prominent? High humidity and warm climates, places that are not clean, like playgrounds, pools, and gyms. There were two methods used for testing the data needed to determine the fungal species and pattern for this study. The tests were the potassium hydroxide mount test and culture testing. The most effective of the two tests was the potassium hydroxide test. Skin scrapings were collected from the active lesions and placed on a glass slide with a drop of 10% KOH solution. This helps dissolve keratin and makes the fungal elements visible under a microscope. The presence of fungal hyphae confirmed a dermatophyte infection, contributing directly to the clinical and diagnostic data shown in the next slide. On this slide, I have provided several different graphs that describe how the data was collected. The first graph is the KOH mount test showing that 47% of the samples were positive, meaning that the fungal species was detected in those tests. Next is the distribution of clinical patterns according to gender. The shape of the species is reflected here on X, and the amount of data compiled for each species that affects either males or females is here on Y. This graph shows very high results for TC annular. Graph number 3 is the organism isolated graph, and it shows how much of each species had been collected. The largest area in purple has no growth, but the next largest in green reflects Trichophyton rubrum. This last graph is the gender percentage graph, and it shows that of 100 participants, 62 were male and 38 were female. The results found in this study were that the most common pattern was annular, and the most common causative agent was Trichophyton rubrum. It's not stated directly in the article, but I'm sure that the research done was used to improve diagnosis and treatments in the future. Interesting facts is that this is a fungus, not a worm or a helminth. This infection has a different name based on where it is in the body, and the word corporis means body. This concludes the end of my presentation. Thank you for watching.